Hello everybody, myself Dr. Vijaykumar, Associate Professor and Head at the Department of Microbiology, School of Science, RK University. I welcome you all to this MOOC course, COVID-19, the pandemic. In today's in today's module, we are going to discuss about the structure and replication of coronavirus. So let us first of all discuss about this coronavirus, what is all about. The coronavirus is a mammalian virus. It is an enveloped virus, which I will be discussing later on. It causes the disease which has been named as COVID-19. So COVID-19 is a disease which is caused by novel coronavirus, which originated in the Wuhan city of China. The coronavirus outbreak, which started in China in the month of December 2019, has spreaded now worldwide, and it has infected more than 189 countries all over the world, and it has killed, as of now, it has killed 280,000 people around the globe, and it has infected around 41 lakh people as of May 10, 2020. The first outbreak of the disease was reported on 31st December in Wuhan city of China. On February, in the month of February, WHO named this causal organism, the novel coronavirus, as COVID-19. Now, you must understand this term COVID-19 because right now you all can hear anywhere this term, but uh, you should be aware and you should know that what COVID-19 stands for. In the COVID-19, you can see the term COVID-19. So the first two alphabets, CO, it represents corona. The third alphabet, that is VI, third and fourth, it represents virus. And the D, it represents the disease. It means the COVID is the disease which is caused by coronavirus. The the numerical digit 19 represents the year in which it was found or it was first reported, that is 2019. That's why the whole term came here, COVID-19. Now, the virus, this is the disease. COVID-19 is the disease, but the virus which causes it, it has been named, earlier it was named as 2019 NCOV, and there is an international committee which actually look after about naming and nomenclature of the viruses. That committee is known as International Committee on Taxonomy of Viruses, and as per them, the internal ICTV, they named the virus as SARS-CoV-2. Now, you might have heard previously the term SARS, SARS. It stands for Severe Acute Respiratory Syndrome. SARS is again a member of, SARS is a disease which is again caused by a coronavirus, which was first reported in 2002. We will discuss later about this, a glimpse of this in the later section. Let us first look about, let us now look about the timeline of the virus. On 31st December 2019, the Wuhan Municipal Health Commission on China reported that among few people they reported that among few people are infected with pneumonia like disease in the Wuhan uh, Hubei province, which was later on even identified. The disease was identified as COVID-19 and the causal organism was identified as SARS-CoV-2. On 1st January 2020, WHO set up an incident management support team which continuously coordinated with the members and with the authentic with officers of China to find more on this, to investigate into the matter. And on 4th January, WHO reported on social media that yes, there was a cluster of pneumonia cases in the Wuhan city, the causal organism of which was not reported by that time. As the time processed, the cases unfolded, and the research also carried on this. And on 5th January, WHO published the first disease outbreak news on the about this new virus. On 10th January, WHO issued a comprehensive package of technical guidelines on its website, which gave and it advised to all the countries that how to detect, how to test, and how to manage the potential cases of this new pneumonia like disease. And all these guidelines were based on what they have observed till that time. And based on the guidelines were also prepared on the basis of their previous experience with SARS and MERS, which had taken which has came up as outbreak in 2002 and 2012. On 12th January 2020, a major breakthrough came into the light that when China reported the genetic sequence of COVID-19. Later on, it helped a lot among the researchers and others to find out to know more about the virus, and to it also helped, and it is still it is helping the researcher community to design and to investigate and design the drugs of against this virus. On 13th January 2020, the officials of WHO confirmed that a case of COVID-19 was found in Thailand, that is the first case outside China. And after this, on 14th January, WHO technical lead technical lead for the response noted in the press briefing that there are chances of human to human communication transmission of these viruses. Before that, they had not talked about human to human transmission. And they also they also mentioned that there is a possibility of outbreak, worldwide outbreak. And on 30th January 2020, the Director General of WHO declared the novel coronavirus outbreak as a public health emergency of international concern, which is the which is the notice of highest level as per WHO. Although it was later they suggested that it is a pandemic, but in the WHO dictionary, PHEIC, that is Public Health Emergency of International Concern, is the highest order to measure the severity of any infection. Since the conceptualization of WHO in 2005, this was the sixth time that WHO had declared any disease as PHEIC. As of now, and you people are knowing every day, you people are aware of that as of now, the whole world has came into the grips of this coronavirus infection. And as I told earlier that uh, today when we are discussing, I have the data in front of your screen of yesterday, that is 10th May 2020, that 40 lakh 41,520 people were infected with the disease, confirmed cases, and globally 27 2 lakh 79,593 deaths have been reported as of now, and this is not the end. It is increasing every minute, every day at a very fast rate, which is currently a matter of concern for all countries, whether it is developed country or it is developing country. Let us go for the classification of COVID-19. The coronavirus has this 2019 nCoV has been categorized into the family Coronaviridae, genus Beta coronavirus, and subgenus Cervicoviruses. It is This virus is an enveloped virus. This virus is an enveloped animal virus which has uh, various hosts, whether it is bird, whether it be mammals, uh, and the enveloped viruses are very much infectious compared to other because the envelope is of host origin, and this helps them a lot to infect the host very fastly. Coronaviruses are a large family of enveloped viruses. They have RNA as their genetic material, single-stranded positive sense RNA as their genetic material. In humans, they can cause mild infection in the upper respiratory tract, but they cause severe infection in the lower respiratory tract. These infections can manifest as bronchitis, pneumonia, or severe respiratory illness such as severe acute respiratory syndrome or mild Middle East respiratory syndrome as it was observed in the case of MERS, or as we are observing as of now, that is the COVID-19 disease. The SARS coronavirus that causes, let us, I want to come, I will give you a glimpse of comparison of SARS, MERS, and COVID-19. You can go through the summary, like the SARS coronavirus, which had been reported in the Guangdong province of China in November 2002. It had the mortality rate of 9.6%. The transmission was as similar to this one, the present case, that is droplet infection, airborne infection. It was spreading through droplets produced through coughing, sneezing, talking, breathing. The minimum incubation period of the virus was 5 days in case of SARS, and the key symptoms of the disease was a cough, dry and the first and later on with cough, a fever, diarrhea in the first or the second week of illness or both. And the risk group were the people who had compromised immune system. There was no treatment of the SARS. There was no vaccine. As of now, there is no vaccine for SARS. In case of MERS also, that is the Middle East Respiratory Syndrome, again the pathogen was named MERS-CoV. Mode of transmission was same, droplet. Key symptoms were fever, cough, shortness of breath. A risk group were people at the age of 60 and particularly those underlying the medical conditions like diabetes, high blood pressure and kidney failure. Again, MERS had also no treatment and no vaccine. Coming to the existing one, that is COVID-19, it has the mortality rate found to be between 1.38 to 3.4 in majority of the cases. In some countries it is more also. The mode of transmission is common as of for the SARS and MERS. Incubation period in case of COVID-19 is 7 to 14 days, depending upon the situation of the individual. The key symptoms are again the dry cough, continuous fever for 3 to 4 days, shortness of breath, and the risk group are. It is said that the risk group is the people below the age of children below age of 16 and people below above the age of 65. But the data which is coming from different countries is indicating that it is infecting equally or almost to each and every age group irrespective of gender, whether it is male or female. But yes, the one common majority have reported the common thing, that is they all or majority have some other illnesses also associated with COVID-19. Treatment as of now, there is no treatment, although uh, few drugs which like hydroxychloroquine, which is used for the treatment of malaria, it has been recommended. And recently the antiviral drug which was used and developed for Ebola, that has been used, that has been recommended by few countries to use as a treatment. Vaccine as of now, no vaccine has been developed for now, but yes, the various pharma companies are in race and they are working day and night continuously to develop, and we can expect, no one can assure, but we can expect that the vaccine may come in the time of 1.5 or 2 years, but it is not sure, because as in the case of SARS and MERS, still there is no vaccine. It may remain the same in case of COVID-19. The pathogenic coronavirus, it has a huge list of important pathogenic viruses. As you can see on your screen, It has the genus, four genuses: alpha, beta, gamma, and delta. And the host, you can see, it ranges from human to pig, dog, camel, cat, then birds also, chicken, whale, mouse. These are all the diverse hosts in which the virus survives. And examples are in front of your screen. These are the various members, these are the different members of coronaviruses which are pathogenic and which poses a danger for the human life, because but coronavirus has always found zoonotic transmission, that is from animal to human, and these viruses mutate at very fast rate. That's why it is not easy to develop a vaccine against those viruses who mutate a very fast rate. And the second thing is, since they are enveloped viruses, that also raises difficulties for developing vaccines against them. Coming to the structure of coronavirus, as I told earlier, it is an enveloped viruses. It has a lipid. The envelope is the lipid bilayer, which is of host origin. The host means the cell which get infected by the virus. When the virus enters into the cell, that cell becomes its host. And when the virus comes out of that cell, it takes a coating of the cell membrane, host cell membrane, onto it, and thus it makes the envelope. It has positive sense single-stranded RNA as its genomic material, which ranges between 26 to 36 kilobase pairs. Now, what does it mean, the positive sense? The positive sense means that the genomic material, that is a single-stranded RNA, is ready to be translated and give rise to various viral proteins. In the case of viruses, it has been found the genomic material can be positive sense RNA, as in the case of coronaviruses. It can also be negative sense RNA, where the RNA itself is not ready to be translated, but before that, it must be converted into positive sense RNA. So, this is the difference between negative sense RNA and positive sense RNA. Now, it generates this the 26 to 32 kilobase pair genomic material, that positive sense RNA. It produces around 15 to 16 non-structural proteins. That means those proteins which are not the part of the structure of virus. I hope you all might be aware that the structure of viruses consists of capsomers, protomers, then it has the envelope. There are various glycoproteins embedded into the envelope. So these are all the structural proteins which gives the virus a structure. But there are other proteins also which do not constitute in the structure of virus. Those are called the non-structural proteins. And such non-structural proteins, the genome codes around 15 to 16 non-structural proteins. I have given the list in the next slide. The 3' end of coronavirus genome encodes 4 major structural proteins. Now you see those which consist of the structure. And these structural proteins are the spike protein, the nucleocapsid protein, the membrane protein, and the envelope protein. Here you can see in this image that the nucleocapsid protein, here you can see the various functions of these structural proteins. The nucleocapsid protein is represented by N, the spike protein is represented by S, the envelope protein is represented by E, and the membrane protein is represented by M. The nucleocapsid proteins are those proteins which surrounds the nucleic acid, that is here the positive sense RNA. So those proteins which surrounds and which protects the nucleic acid are known as nucleocapsid proteins. In this image, you can see the structural proteins of the virus. As you can see here, the spike glycoprotein is here. The hemagglutinin acetylesterases, it is again a protein embedded into the membrane. The nucleocapsid phosphoproteins, the viral envelope, which is of host origin, and the viral RNA, which is a positive sense RNA. These are the list of 16 non-structural proteins which has been identified as of now. I will provide the reference article which you are seeing here, the references. I will provide 2 to 3 research articles through which you can get more about these non-structural proteins and their functions. Let us now come to the virus replication. After uh, this will give you a better understanding of how the virus replicates into the host, and how and why it is so dangerous, and how it can also give you an idea about what can be the target sites for controlling the virus. In the first image, in this image you can see that it it indicates the various steps of virus entry and replication. In the very first stage, the virus attaches to the surface receptors. The surface receptors are present on the surface of the host cell. Here, in case of coronavirus, it is ACE2, which is also known as or stands for angiotensin converting enzyme 2. So ACE2 is the receptor on which the spike glycoprotein of virus attaches. In the second, here you can see when the virus attaches with its spike to the ACE2, the envelope get immersed or get mixed get merged into the membrane of the host, and the genomic material is released. Now, when the genomic material, that is positive sense RNA, is released, from here it goes for two processes goes simultaneously. One is the translation, because the genomic material of host is divided into early phase proteins and late phase proteins. Few proteins which are needed for initiating the replication of the viral genome itself, and few proteins which are required at the later stage of the replication for assembly of the complete virion. So in the first phase, when the positive sense RNA is translated, it codes for the early proteins. These early proteins codes for makes the enzymes which are required in the later stage for reverse transcription of the positive sense RNA. Now, why it is required? Because the RNA, it is positive sense RNA, so it must be converted into cDNA, from which the copies which will be generated, the mRNAs which will be generated from the cDNA, they will be again the positive sense copies, and these positive sense copies will be packed into individual virion, making it a complete virus structure. So in this, you can see here, these it represents the open reading frames. These are the, you can see here, the translation represents the translation of different frames codes for different, like S protein, that is spike protein, NSP, then E, then M. These are the different proteins which are coded by the uh, mRNA, positive sense mRNA of the virus. On the other hand, the latter phase, these are the latter proteins also which are coded by them, and at the time of packaging of the virus, they get embedded into this envelope, and thus, and you can see here that previously these are arranged on the rough endoplasmic reticulum. After that, from the rough endoplasmic reticulum, the vesicles are formed which merge into the Golgi. From Golgi, the complete vesicle containing the active virion, then it gets released from the cells with the help of the process known as exocytosis. So this is how the virus enters into the host, replicates its genomic material, produces the viral proteins, and then exits from the host. Second diagram here, it gives the more detail about the same thing which I have told here. You can see here, it also gives you about the different targets where this virus can be controlled, where different enzymes which can be blocked, and thus the process can be stopped. Like you can see here, chloropromazine, which can stop the endocytosis process, and similarly there are many other targeted targets. This target will be the targets for the drug. This becomes the target for drug development, and it will be discussed into another module, which will be taken by Dr. Rajesh Patel. So I hope you have learned a lot in this session. Now go through the MCQ and check how much you have obtained from this session. Thank you for listening this. Thank you very much.